Hello everyone, and thank you for listening to my talk today. My name is Rene Warren from BC Cancer Genome Sciences Center in Vancouver, Canada, and today I'll be talking about SARS-CoV-2 variant time maps. Needless to say that COVID-19 is a dire situation with millions of lives lost and a toll on human health, impacting all sectors of human activity as well, with a grave socio-economic impact that will be felt for years. The agent of COVID-19 is SARS-CoV-2, a coronavirus related to SARS-CoV, which has a compact 30 kb RNA genome of 10 genes, one of which encodes a surface glycoprotein or spike protein, which binds host ACE2 receptors to gain cellular entry. It is airborne and contagious. On the right side, you see a three-way sequence comparison between SARS-CoV on the left, SARS-CoV-2 on the top, and MERS-CoV on the right. And you can see high sequence similarity between the SARS-CoV and SARS-CoV-2 genomes. There are global efforts to sequence millions of SARS-CoV-2 genomes. These are spearheaded by medical clinical sequencing centers and public health labs worldwide, who then submit their genomes to central repositories such as the GISAID Viral Genome Repository or the Sequence Read Archive. There are other initiatives such as the Canadian COVID Genomics Network, CanCoGen, with the aim of sequencing over 10,000 COVID-19 patient host genomes and associated SARS-CoV-2 genomes. Groups worldwide are using these resources to track a changing virus. They offer great intuitive user interface, which are largely based on phylogenetic analysis of a limited sample size, and they don't always provide the raw variant data. What we wanted to do with this work was track the evolution of SARS-CoV-2 genomes in human hosts and provide a variant's bird's eye view and answer specific questions such as what distinct single nucleotide variants, how many, how fast do they accumulate, when did they emerge, and where. To achieve this, we built a SARS-CoV-2 variant analysis and dissemination pipeline that consists of 4 steps, which begins with a GISAID genome download, followed with variant profiling with NT-edit, plotting with scalable vector graphics, and web hosting of the resulting maps at bcgsc.github.io/SARS2. SARS-CoV-2 genomes download is done by accessing the GISAID portal, specifying complete high-coverage genomes, which are derived from samples acquired at specific dates. Now for the variant profiling portion of the pipeline. Because GISAID genomes are updated frequently, we process SARS-CoV-2 GISAID genomes daily. It's an automated process in which we iterate through each GISAID genome and generate Bloom filters with NT-hits. These Bloom filter data structures are then read by NT-edit for a single nucleotide variant calling against a reference Wuhan human 1 coronavirus isolate, first sequence early in January 2020. We then parse resulting VCF files and summarize each GISAID ID as shown below. Additional scripts map the effect of nucleotide variations globally. In summary, we've processed over 1.5 million complete SARS-CoV-2 GISAID genomes from 6 continents since January 2020 and identified over 30,000 distinct nucleotide variants observed in 10 or more genomes, and identified as high as 46 SNVs per genome. Overall, we observed that over 21,000 discrete sites are mutated in the 30 kb genome. So that's a little bit over 72%. And identify 626 convergent mutations. There's an example shown below for the S gene, where 2 nucleotide variants yield the same missense variation, W152R, in the S protein. All SNVs are then plotted using a comprehensive high-resolution output. For this, we use scalable vector graphics. On the map, variants are organized along the genome axis. Each years are arranged clockwise, where 1 radius equals 1 day. Each SNV is a spot on those maps and colored by region and sized by sample count or ratio. The maps are fully interactive with tooltip, zoom, pan, drag, tilt, and highlight. Mouse hover reveals the date, variant, regions, count, proportion, gene, product effect. Once all the SVG's time maps have been generated, we host them at bcgsc.github.io/SARS-2. We host over 120 maps in total. Each map comprises a wealth of information that can be mined to gain further insights. For instance, the map on the left shows, for year 2020, missense variants on the S genes in all of Europe, with 2 regions highlighted. These regions are zoomed in on the right side. In the bottom, what we see is the emergence of the N501Y variant in Wales, UK. And on top, near the end of the year, at a much higher frequency in England, UK. The information we provide can be used to generate animations such as this one, which show emergence of variants daily. Raw SNV data can also be used to generate plots such as this one that shows the normalized change in abundance weekly. This allows the identification of specific variants. You can find more detail on this work by accessing our publication and bcgsc.github.io/SARS2. I'd like to thank our funders, NIH, Genome BC, Genome Canada, and the John Jamber Education Fund. Thank you very much for listening and for your interest in our work. I'm happy to take any questions you may have. And this is our poster.